Hello again, and welcome to another episode of Max Planck Florida's Neurotransmissions podcast. I'm Joe Schumacher, joined, as always, by Lesley Colgan. How you doing, Lesley?
Great.
It's great to see you. We've got a really interesting episode today. How's your social competence these days? How are you feeling? Are you socially competent?
Oh, that's a tough one. It depends, sometimes on the situation, I think, and whether I've had a glass of wine to drink. Maybe.
Okay, fair. So our guest today is a renowned expert on studying the neural basis of different types of social interactions in the brain. Maybe before we talk about her and her research, let's define social competence. What would you say is, based on everything we've learned over the last couple of days, a good operating definition of social competence?
Yeah, I think from what I understand, social competence is the idea that, you change your social behavior depending on your context, right? So you're not going to necessarily act the same way with your children or your best friend as you would with your boss, for example.
Right. If I go to a baseball game, I might be behaving in one particular way. If I'm in a meeting with our CEO, I might behave a little bit differently, unless we're both at a baseball game. David, I would love to go to a baseball game with you if you're out there. Anyway, our guest today is Professor Nancy Padilla-Coreano, an old friend of mine from graduate school days. She is an assistant professor in the Department of Neuroscience at the University of Florida College of Medicine. She's a recipient of many awards and accolades, including a Brain Initiative K99 and R00 Pathway to Independence Award, the Henry Grass Neuroscience Rising Star Award, and the prestigious L'Oreal USA Women in Science Fellow. Dr. Padilla-Coreano is an expert, particularly when it comes to understanding the neural mechanisms governing the control of social rank, and establishing basically what are the coding mechanisms that distinguish mice at different parts of a social hierarchy from each other. So in a social hierarchy, you might have some very dominant mice that are very prone to overruling the behaviors of others, and then subordinate mice who kind of just fall in line and behave in accordance to those dominant ones. She's done some really excellent work, particularly in her postdoc and now in her early days as an assistant professor, to understand parts of the brain that control that and ways to manipulate them.
Yeah, she's really sort of a pioneer in this whole sort of expansion, I'd say, in the field of neuroscience, of really understanding social neuroscience behavior. And another thing I think we should point out about Dr. Padilla-Coreano is that she has co-founded a podcast, Stories of Women in Neuroscience. It's actually a whole platform, a podcast, as well as a website that really highlights women in neuroscience and has a lot of resources that are available to everyone at all stages of their career. And it's a really wonderful platform. So definitely check that out, Stories of Women in Neuroscience. And
we don't get into too much detail about this. We talk about it a little bit in our interview. But one of the things that I think we should take notes on is not only has the Stories of Women in Neuroscience sort of media enterprise grown in a way that it's got both written and podcast related content, but it's also a community building tool where people from all over the world are contributing essays about particular women scientists in the neurosciences. It seems like a really great opportunity to get people at all training levels with lots of different interests in neuroscience involved and part of a community. And I think, that's a real strength of this website. So please check it out, Stories of Women in Neuroscience. It's out there. So without further ado, here's our interview with Nancy Padilla-Coreano. Hi, everyone. Welcome back to another episode. Today we are joined by Dr. Nancy Padilla-Coreano. She's an assistant professor in the Department of Neuroscience at the University of Florida College of Medicine. Dr. Padilla-Coreano, thank you so much for joining us today. And it's fantastic to see you because we actually know each other for a very long time from grad school days. So it's great to see you again.
Yeah, Joe was I don't know if you remember this, but you were my assigned buddy.
I was your buddy. Yeah, I know. Early on at Columbia, they kind of did this matchmaking thing. It was like, this person likes systems neuroscience, this person likes systems neuroscience. And Dr. Padilla-Coreano.
You can call me Nancy.
I know, but this is the public.
Okay.
Nancy was my buddy in grad school, and so I remember a few conversations, like helping you think about thinking about which labs you were interested in rotating in, and that sort of thing. Of course, I remember those days. Ultimately, you ended up joining the lab of Dr. Josh Gordon, who is now the current director of the National Institute of Mental Health, I believe.
Yeah.
So at the time when you were joining was the star power like super obvious with Josh?
He inspired me, but I didn't think he was going to become such a powerful person, to be honest.
Yeah.
Yeah.
Do you guys still keep in touch?
Oh, yeah.
Yeah.
Yeah, we keep in touch.
That's
yeah.
cool. And is he still running his own lab now that he's in
Yes,
this
but
position?
he hired like a scientist person that, a very amazing scientist that's the de facto, like assistant professor of the lab, basically, because his day-to-day is pretty busy. But no, we stay in touch. He just visited UF, gave a big time seminar, but he said that what he wanted was to see my lab.
That's
so
cool.
it was
That's awesome.
it was really awesome.
That's
yeah.
really cool.
Yeah.
Well, Josh, if you're out there, we'd love to get you on the podcast someday. Anyway, well, thank you for joining us on the podcast. I think one thing that Lesley and I were really interested in talking about is that you actually have your own podcast that has been going for a while now. Tell us about stories of women in neuroscience.
Yeah, so it's definitely like a team's effort. The idea, I had the idea first in the sense like I was like, oh, what if we did this? But it was only became a possibility and a reality because I found people that were like equally excited about the idea. But it's a multimedia project because we have episodes that feature different women neuroscientists at different career stages that do different types of neuroscience, that come from different backgrounds, that come now from different parts of the world because we have some team members in the UK and in France that can cover other time zones better when interviewing people in their 9-to-5 life. And so every episode features a woman. And then with that episode, there's a written component that goes into our website where we create a profile. And that's a little bit more geared to like an undergrad with no neuroscience background or with a little neuroscience background can understand that written profile. So that's a little bit more like general public science communication. And then, we try to gear the interviews for everybody to understand, but sometimes we go to the nitty gritty in the actual interview. Yeah. So we have a podcast and profiles online.
Yeah, and I love that on the website, you also have a ton of really great resources in terms of, grants that are available and, discussions of issues such as intersectionality and things like
that's
that.
true. Through the years, because it's such a passionate group of volunteers that's behind the project, the behind the scene is like, it's huge, especially because it's all volunteer based, so everybody does what they can. But there's lots of passion about like, what can we do for the community? Because sometimes we get emails. We have an email account, and we sometimes get emails for career advice. So that kind of created like, perhaps our website could have some resources. So that's where the resource page came from.
Yeah, that's really great. And I love that one of your missions is sort of, increasing the transparency behind, these amazing women in neuroscience and sort of telling their career journeys. And so I thought now that you're a guest on our podcast, we could kind of turn the tables a little bit and actually ask you a little bit about your career journey and your unique path into science.
Yeah, that sounds great. I'm excited.
So what was it that first sort of got you interested in science? I understand you were actually a musician when you were younger.
Yeah, so I went to this music school in Puerto Rico. It's called Escuela Libre de Música. And it's a public school that's specialized for music. And my mom's a music teacher. And so I grew up surrounded by music, and I went to this school, and I'm a good student. You put me on an environment where you're telling me learn something, and I like go in and learn and really immerse myself in it. And I really thought I was going to be a musician like my mom. And but towards I think 9th, 10th grade, I started I was like, I just want to understand why music is how it is, like how come it changes our emotions? How come it changes how we feel, and how come we can memorize songs better than words without rhythm? So I just started having all these questions and these crazy theories like, oh, I bet I could treat people's brain disorders with music. Just like some really out there ideas. And like I was I think young me was much more like the sky's the limit than even now. So I was like, yeah, I'm going to be a neurologist. And also I was inspired by Oliver Sacks. Like, so I'm going to be a neurologist just like Oliver Sacks. And I'll do experiments with music with my patients. And that was sort of like my life plan in high school. Then nobody in my family had done like any STEM careers really. So I was like, like you want to study the brain, then I'll become a neurologist. I didn't know what neuroscience was. And then in undergrad, I encountered that there were neuroscientists, and very quickly I shifted to the PhD track once I experienced lab.
So, I mean, you mentioned Oliver Sacks. So you were actually at Columbia at the same time as Oliver Sacks, I think.
I didn't know that, though. I knew he lived in New York City, but I was never able to see him in person, unfortunately.
I think he was like, some kind of emeritus situation or something
Makes
like that
sense.
when we were there. But yeah, I was curious if you'd met him. But
unfortunately, I didn't. No. But he inspired me anyway.
So what brought you to Columbia?
It was, like when you're applying to grad school, you like, at least I focused on, does this program have enough like people I'm interested in rotating with? So there were, I would say like a handful of schools, like 3 or 4, that all of them like had like, I was interested in, like at that time, neural circuits was like, I want to get into neural circuits, and like I want to do electrophysiology in vivo. So those were the things that I was paying attention to. And funnily enough, even though I had visited New York City with my family and actually hated it, I was like, oh my God, this is so overwhelming. But I think deep down, I'm a very social person, and I knew people in New York City. So I was like, I really like the research there. I had met some of the grad students, including people in Josh Gordon's lab, and I was like, I kind of see myself there. People in other labs too. And so I had a really good feeling about the possible PIs I could work with, and I knew people. And that was just enough for me. I was going to have an instant community. Even though it was an overwhelming city, I was like, I can have, do things on weekends with people that I already know. Some of my friends that were musicians had actually established themselves in New York. I ended up hanging out less with them because their schedule was more like go out at midnight until really late. But some of them even came to my defense.
That's cool.
So that was really cool. Yeah.
Okay. Well, speaking of social, I guess this is the best segue I can do. The work that you've done since your postdoc has really been focused on social competence, social dominance hierarchies, and how that's sort of established in the brain. So, coming from, I believe you were working on sort of like hippocampal prefrontal cortical stuff in a grad student. What got you interested more in the sort of functional behavioral, like almost like ethologically behaviorally relevant stuff that you did as a postdoc? It
was a really interesting like little path. So at first, at the end of my PhD, I remember thinking I felt unsatisfied with the behavior level I was measuring. I had when I reflected on it, I was like, wow, I use this maze, and this is like the unidimensional measurement of behavior that I have, the location of the animal. And so I wanted to get more into, most of the interviews I did was like considering labs that did hypothalamic behaviors, so like aggression or like mating or things that look like way more complex and people weren't measuring with at least there were multiple animals in it. So social behaviors, that's where I first encountered the possibility of studying social behaviors. From the more I want to go into innate behaviors. I was thinking I was going to go into innate behaviors, which had aggression and mating. And then I interviewed with Kay Tai, and honestly, up until I interviewed with Kay Tai and read her one paper that they had where they reported that social rank affected these other, behaviors of the animal. And I was like, what? mice have social ranks. So anyway, I decided to join that lab. And when I had choices of where to go, like that was the direction of like, I just wanted to understand what was even social rank and what were these hierarchies about. Because it seemed like it was something fundamental that was complex enough behaviorally, which is one of the things I wanted, but it like very important to understand from my perspective. And it happened to overlap with like some of the brain regions that I had been studied before because I was studying the hippocampal prefrontal projection. So I was like, oh, at least I can, there's like something that I know, which is the prefrontal cortex that seems to relate to this. So that gave me like an entry point of comfort.
Right.
Yeah.
So because I have a similar question, which is related to mice have social hierarchies. Mice have sort of established dominance hierarchies. Like from an evolutionary perspective, like it seems interesting that, and you talked about this in your talk today, so many different species establish these complex social networks. You showed the picture of the Japanese baboons like chilling in the hot tub, and the mice and all these things. Humans, obviously. What sort of purpose do the different types of social hierarchies that mice have serve them, like just behaviorally in the wild?
Yeah. So the first one is reducing aggression, conserving energy, like and then sharing resources and territory effectively. So those I would say like those are the main drivers for it. There's also for males, it seems like it's an advantage to be dominant because then you'll be able to mate more because females are more attracted to the smells of dominant males. So there's that evolutionary advantage too of like you get to pass your genes more.
So the conservation of energy thing is just like, as long as there's kind of like a subliminal policing system that keeps everybody in order, it means that less people are constantly trying to attack each other to
there's less
to
fighting for the resources because there's like, so actually in primates this is like huge in terms of I love going to conferences and talking to scientists that work with different animal species. And last year I went to this conference where there were a lot of primate researchers, and we were talking about my research because I had just given a talk, and they're like, oh, let me tell you these like funny stories about how much our primates like care about hierarchy, even though we don't study that. When they give out the treats in their animal facility, they have to go in order of dominance. If they violate that dominance order, they all start screaming.
That's fascinating.
Yeah.
When I was in grad school, I read a book by Robert Sapolsky. This is an anecdote with no point. We might cut this. But like, it's a book called The Primate's Memoir by Robert Sapolsky.
I know that book, yeah.
It's a fascinating book, really amazing book. And he was sort of talking about in baboons, all this field work that was being done to basically understand like troops of baboons and how they work. So are there similar levels of understanding of how rodent species establish their hierarchies as people have studied in primates, or is this a new field, like basically?
You mean like in the wild?
Or in the laboratory, just like how, like the rules behind establishing social hierarchies in mice, is this something that is a new area of research, or is it pretty well characterized?
Honestly, like, okay, so there's work from like the 80s of some people. Jane Hertz, she's an investigator in the UK, and I think this was like her PhD work. It's like observing these group of wild mice in a barn, and she made these amazing observations of that group. So I don't know, like, so there's clearly like, maybe this is not exactly ruled wise, but it was clear that the territories related to where the food was, and that like both females and males like were guarding like the territories that were like had the food, and the higher the rank, the more likely you were to mate with the higher ranking female and males, and more access to the food basically. So
pretty well established.
So at least from her work, that is established. But this is wild mice, and like we study like these mice that are not exactly the same. So, and a big challenge is that in the lab setting we don't really have them in female male groups, right? So they're in our lab when we study males, their drive is purely territorial rather than like for reproductive in the context that we study it.
So when we think about sort of trying to study these really complex natural behaviors in the lab, I think it's fair to say that there are a lot of challenges.
Yes.
And I think this whole field of social neuroscience has really sort of exploded over the last, I don't know, maybe 5 to 10 years. And I think a large part of that is the development of sort of more advanced technologies. And you've been sort of part of the forefront of that. And so I was wondering if you could, talk us through maybe what some of the challenges are in trying to replicate and study some of these social behaviors in the lab.
Absolutely, yeah. So to echo what you said, I agree. Like, I think it's because there's been these technological advances that allow us to study social behaviors more easily. Like, so in the last 5 years or even less, like deep neural networks have gotten good enough to do what people call multi-agent tracking, right? Multi-animal tracking. And that's just been like opening an avenue of like, oh, we don't have to do everything by hand. We could quantify this with more precision than just having a person like annotate every single frame. And so some of the challenges that exist with all social behaviors are pretty similar to the challenges that exist with any behavioral neuroscience phenomena, which is like the conditions, the housing conditions, the baseline conditions of the animals really change a lot of things. For example, more stress. If your housing facility for some reason is like has higher levels of stress, then the hierarchies are less stable and things like that. So anything that affects animal behavior, it's going to affect your whole group. So, and another thing that's a challenge so one of my lab's first study is actually like a systematic review of the literature. And, we analyze the methods of 700 papers roughly. And of that, we're studying social memory because I wanted to understand what type of social memories have been studied and how have they been studied. And one major challenge that we found, and this adds to like the challenges of reproducing things, is that, when you do social experiments, you actually have more than one animal, right? The subject and some other animal that's like part of their interaction. Turns out that many studies don't report very little information about the social stimulus. But from all my work, like in social hierarchies, I know that who the other is matters a lot. So then that's one challenge, like if you're just treating another animal as a very unidimensional stimulus, you're not even reporting their sex, their strain, their age. Are they living alone or are they grouped out? So like all these things that affect the behavior of that animal and therefore are going to affect the behavior of the subject, but it's not even documented. It's a huge challenge.
Yeah, I can see that. Or social rank of the animal, for example.
Yeah. Three papers out of 700 plus consider or even like talked about social rank. So
really interesting. So, maybe we could just step back a little and define some of these terms for our audience a little clearer. So we talk about social rank. What does that mean? And in the lab, how do you actually determine what social rank, what animal has what social rank?
Yeah, so the social rank of an animal is their position in a hierarchy, right? So like the higher the rank, the more dominant they are. So you can imagine you have a group of 4 mice. You have like rank number 1, that's your most dominant animal. Ranks number 2 and 3, those are your intermediates. And then rank number 4. And what do I mean by dominance is like, this is how the behaviors in which they're consistently winning in points of conflict, in points of social competition. So how do we measure that in the lab? The most basic way, which is really, really hard, is to observe every single interaction they have in their cage. But that is incredibly hard. Although investigators like James Curley take that approach by having these like giant like cages where they can do observations of large groups of animals that are undisturbed. But in labs where we're doing lots of manipulations and lots of behavioral measurements, we take the group out, right? We disrupt the group constantly when we take them out of the cage. So we're changing their normal group dynamics. So then there's other ways to read out the hierarchy, basically. Popular test is they're based on their territorial urine marking. You know how dogs mark territory? Mice do that too. So if you put them in this clean environment and you put two of them and you put a divider wall where they can smell each other, because it's a new environment, they're going to start marking the territory. And because there's another animal present that they can smell, if one of them is subordinate to the other, they'll mark less territory. They want to pull their pee in a corner so that they don't bother the dominant one. Versus the dominant one marks freely. So looking at those patterns of urination is actually a way of knowing their dominance. That's one of them. That's very ethologically based because, they are territorial. And then there's others that are more popular, like the tube test, which is this narrow hallway of a tube that you put two mice together. They encounter each other, and they cannot cross each other, so one of them pushes out the other. And that's another readout of dominance.
So that actually gets to something I thought was really interesting about that you kind of said very upfront in your talk was one of the challenges with these really fluid, constantly evolving behaviors is that they have no trial structure to them, really. Things like the tube test or another test that you implement in the lab, like allow you to have like on a trial by trial basis outcomes, like who outcompeted the other.
Yeah.
It allows you to sort of in real time keep track of these things. And that's important, I think, in studies like what you've done, which is to try to understand parts of the brain involved in encoding social rank. So what are some of the other ways that you have mice kind of competing against each other in a way that emphasizes their rank, and how do you use those to understand which parts of the brain are involved in mediating those types of interactions?
Yeah, so during my postdoc, I came up with an assay that's trial-based that imposed some of the structure that's unnatural to the mouse, but convenient to the investigator. So we especially to do electrophysiology, to record from neurons and look at what they're signaling. And so basically I took a tone and paired it with a small drop of reward, and we individually train each mouse from a group to associate that tone and that reward. And once every animal in the group knows that tone means that a reward is coming, then we start pinning them together, like putting both of them in the presence of the tone and then seeing who gets the reward. And then that tone is really organizing their behavior because it signals when the reward is coming, when do they have to compete. So then that way we could record throughout the session that all these tones are playing randomly and then see the responses to the tone. So it gives us a structure.
And so simultaneously then you can also record the activities of like many single neurons in the brain using electrophysiology.
Exactly.
Say you're, brand new, you don't know exactly. The brain is a very complex area. You focused in on medial prefrontal cortex as a region of interest for studying this. Why that region specifically? What was known about it before you started understanding its role in establishing these social ranks?
Yeah, so there were two studies that really motivated me to focus on the medial prefrontal cortex. But before mentioning those two studies, just generally, like the medial prefrontal cortex and prefrontal cortex, especially in the primate and across species, is super important for decision making. And integrating information and kind of like goal-oriented behavior. And, there's some theories about the prefrontal cortex that it holds information with enough time for the animal to like do their goal-oriented behavior. So it's like some people say that, it has like rule-related information or goal-related information rather than just stimulus-driven. So it doesn't just reflect the sensory stimulus, but it reflects the sensory stimulus in consideration of the rules or the context or internal state of the animal or something like that. So that's the general background of the prefrontal cortex. And there were two particular studies that really narrowed it for me to the medial prefrontal cortex, and one of them is in humans. In the case of humans, it's really medial rostral PFC, and they showed that as people were engaging on this game where they had to report the ranks of the players, so this is like a hierarchy of gamers in this particular game for this experiment. As they learned the ranks and reported, this individual is higher ranking than this one. That learning correlated with activity of the medial prefrontal cortex. And if they use transmagnetic stimulation paired up with them doing the task, they will learn the social ranks quicker. So, that's very artificial because humans, they're not really living in the context of like those ranks really mattering. But then this was reinforced by a study in rodents, where in the natural structure, quote unquote, for a lab mouse, their cage mates, that's their natural hierarchy. That's the hierarchy that matters to them, because they experience aggression and share territory with those animals. In the context of that hierarchy, if you stimulate opgenetically, with a way to like manipulate specific neurons in the brain, if you stimulate the neurons of the MPFC, you can actually change the dominance behavior. And if you stimulate strong enough, you can even change the rank permanently.
So you were, recording in the medial prefrontal cortex based on, this information that might have something to do with social dominance or social rank. And while these animals were actually performing this competition task, and you had, mice of maybe two different dominants, a dominant and a subordinate mouse. So what did you actually see in the recordings?
Yeah, so I found a few interesting things. So one of them was that the way that the neurons in the prefrontal cortex were responding to the things that were happening during the competition, like whether they were getting their reward or not getting their reward, or whether they were picking up their reward, or when the tone was first playing, the way the neurons responded to those things depended on the rank of the animal. So the rank at baseline was affecting how those responses in the brain. And then another thing that I found was like if you looked at the activity of all the neurons as a population, you could actually predict just from looking at the activity of the population what was the rank of the animal. So it seems like the social rank of the animal perhaps is represented as a rule with sustained activity in the prefrontal cortex for the animal to guide its behavior. And then we finalized our study by like identifying a specific output population of the prefrontal cortex that goes into the hypothalamus, that if we manipulate those neurons, then we see changes in who wins, basically.
Basically, I guess what you're saying is that for these, I think they were they lateral hippocampus?
Yeah, lateral hypothalamus.
Lateral hypothalamus neurons, when you stimulate those neurons in the subordinate mice, they're more likely to kind of assert themselves to win this competition. Is that right? So one thing, the idea of decoding the animal's rank from the neural activity, which was like really interesting aspect of your talk today. One thing that really struck me was that you can predict the outcome of not just the outcome of the trial, but the identity or the rank of the animal many tens of seconds before and after the actual event occurs where they actually have to compete. So what are some of the implications of that? What does that mean about when these two animals are just in a room together? Does that mean that the baseline activity patterns in a dominant and subordinate mouse are just distinct enough that like that's kind of established at the level of that part of the brain in both of those mice?
That is exactly the implication. The implication is that at baseline, at the population level, these patterns of activity are distinct enough that you can tell from the patterns of activity whether they're dominant or subordinate constantly, basically. So,
Lesley is obviously this like type A domineering personality, always talking over me in the podcast setting, and I'm this humble subordinate podcast host. So we have these different brain states in our brains right now, obviously.
Based on my study,
what could somebody like me do to sort of fix myself? Like, how do I become like a little bit more of the person who's going to go and get the juice? Like, that's really the question I want to know.
Well, what I know, not necessarily from my study, but for the combination of my study and other studies, is that these ranks are flexible. They're not fixed through birth, right? Like if once you're in a different, maybe you switch rooms, that's a different context. And then
maybe it's the chair.
Maybe it's the
chair. Try that next time.
But for the mice, it matters like who they're with, and it's all relative to who they're with. And so you could take a dominant from one group, put them in a new group, and then they might become the subordinate.
Or you could take a bunch of subordinate mice from a bunch of different groups, put them together,
one of them will become dominant, right?
And exhibit the same sort of behavioral phenotype as a normal dominant mouse.
Exactly.
Interesting.
Yeah, so they're flexible. So that means that there's hope for you.
That's really interesting to think about it as sort of like based on your experience and a learned response and something that's flexible. But is there also sort of an innate component to it as well? Are there some genetic components
That's a great question. These particular mice that we study are inbreds, which means that genetically they're pretty similar. They could have some epigenetic differences. So in this particular scenario of these mice that are genetically very similar, we see flexibility. I am sure that if we put a pool of like highly, genetically different animals from the same species, genetics will definitely play a role because it could affect things like their motivation, their size, all of these components. I'm sure of that, although I don't have evidence directly because most of the labs are using inbred mice that don't have genetic variability as much. Yeah.
So I wanted to talk to you a little bit about, you started your faculty position about a year ago now, is that correct?
Yeah.
Yeah, so I just wanted to talk to you a little bit about sort of, one, the science of where your lab's headed, but also sort of your experience as a new faculty. So maybe let's start, if you don't mind, with a little bit about your experience, and then we'll get into sort of the future of your lab. So I sort of feel like there's still a little bit of like mystery behind, what's it like as a brand new faculty. And so I think it's nice to have someone who's just starting in that career stage to sort of talk about their experience. What was it like on your first day when you walked into your lab? Was there something that has been particularly challenging for you as a new faculty that you weren't expecting?
Yeah, I mean, lots of things. Oh my God, it's like a roller coaster of emotions because it's like, this is the job, my dream job. And okay, so the first thing was like, obviously when I got more than one job offer. So you feel very confident when you're signing those documents, like, oh, like this is like finally, like I'm going to start my lab. Then once it becomes really real, first of all, you rely completely on other people. You don't know so many things. You can't hire a person without the help of other people. There's an IACUC, but who's the person you need to email for this thing? Who's the person you need to... So you're completely at the mercy of others, especially a lot of administrative support. And so you have to humble yourself in the sense, like, in this new environment, you don't know much, right? And you need the help of so many people in order to make your lab functional, meaning like at least to get the permits, to hire the people, to buy equipment, right? Like it's not like they put the money on my pocket, right? Like I have to learn how to like use the system. So there's lots of learning. And then one thing I wasn't expecting is that when I made my first mistakes, because like if your lab has it all depends on the composition of your lab, right? Like if your lab has a lot of people that have very little experience, there's a knowledge gap that's really big, where it's been years, like a decade since I've been in their position. So I don't remember exactly what is assumed and what's not. And I can make my best prediction of like, oh, here's the things I definitely have to tell them. And so if I forget to tell them something that ends up being really important, then they have no idea, right? Like, so my mistakes mean if I make a mistake, everybody makes a mistake. Nobody can catch my mistakes. So it's when I was a postdoc, if I was surrounded by experts, especially in a lab that's big and like you have other postdocs with similar expertise. They can catch your mistakes immediately. You're telling them about an experiment, they're like, oh, what about this? What about that? What about that? In a lab with very, very brand new, like fresh, exciting students, in my case, it's mostly undergrads and technicians fresh out of college. So in those post-bac years, I say, this is the exciting experiment, this is our controls, and that's it. They just trust me, so that was so new to me because I had not experienced that, because at every stage of my career, people are just like challenging. So that means that when I make mistakes, everybody makes mistakes. At this point, we're 1 year in and like people are starting to challenge me, which is great. But that was scary. So I think the first moment I made a mistake, there was one thing that wasn't working. And then one random night, I was like, oh my God, I forgot about this thing. I did not tell them. about this thing. That's why it's not working. I solved the mystery. But it was my fault, right? So that first moment that happened, I remember thinking, oh my God, I wasn't ready to be a PI. Like, so, that was a surprise of like, oh wait, imposter syndrome still comes back. Oh yeah, it does. But it's fine. I mean, like, this is part of the growing pains. Like, you're growing together. They're learning and you're learning to be a PI, and that's just part of it. So that was tough. Like, like the mistakes are felt stronger, but also the successes are felt stronger. So in my first year, I mostly had undergrads working in the lab, and a couple of the undergrads that spent the last summer in my lab, they had posters that were presenting at this big conference. It's called Abracams. So a conference for mostly undergrads. And both of them got presentation awards. And when I found out, I just felt so amazing, for the successes of students in my lab. So both the lows and the highs are lower and higher than it was before. So
that's really exciting.
Yeah.
So without spoiling too much, in this first year of being a PI, what have been some of your sort of experimental priorities building off of your postdoc work, and what's sort of on the horizon? What are some of the questions you're really interested in pursuing?
Yeah, I mean, the thing I'm most excited about, because I have like too many ideas and too many projects directions, but the most excited that I am right now is about, like, I've been studying competition for all these years, and it's made me wonder about cooperation. What about cooperation? Like, is that in a continuum? Is it the same circuits and networks, just shifting the animal towards one direction versus the other, depending on the situation? Or is it completely non-overlapping or almost non-overlapping circuits that are inhibiting each other, right? So then we're starting to establish some ways to measure cooperation with the goal of being able to, like, recording both conditions and do the same manipulations and see if they change both behaviors in opposing manners or not. So I would say that's what I'm most excited about.
Mice naturally cooperate in certain, or do
They
you have to train them?
they will not cooperate as much as primates, but they do cooperate. Yeah. So there's a particular assay that was created by a group in Italy. I think Papaleo is the name of the PI, and Chiega is the first author, Diego Chiega. And they created this, like, task. Of course, it's not very natural, so that's the challenge. But it's like the mouse can pick to a choice of rewarding themselves or rewarding both themselves and another animal across a little screen. And most animals prefer to reward both themselves and the other. And then this is changed depending on the familiarity. For strangers, there's less cooperative behavior. Like, so if they're familiar, they'll cooperate more. If the other one is subordinate to them, they'll cooperate more too. So there's certain factors, social factors, that affect that cooperative behavior. So that's how we can measure it easily. But we're hoping, so one other way that we can study it is that foraging behavior, which can be done cooperatively in the sense that the behavior of some animals influences the others. So, like, maybe, we can get creative in, like, but we haven't figured that the creativity part takes longer. So for now, we're starting with these, like, structured tasks. And then we're hoping to get a little bit more ethological with, like, foraging of food.
I have some recollection that mice will sort of collectively raise their young, like, sort of in a big group. Am I wrong about that? Is it usually one mom and many pups, or is it sometimes multiple moms?
I mean, they definitely live in groups, but I don't know much about parenting. So that's a place where I need to consult my colleagues about, like, do they actually, multiple adults to a pup, or and even, like, adults that are not their biological parents. So in the future, I'm like, I might work with spiny mice who do have, like, a family structure, and there's some investigators at UF that already have them, so it makes it more possible. But spiny mice are very social, and for sure they take care of their youngs in like groups, like mom and dad take care of the youngs instead of just like the mom. They're monogamous. But so I don't know about.
Also hard to study from like a trial-based, approach, the way that's, helpful for the experimenter, I suppose.
Yeah, that's so true. Yeah.
Well, Nancy, thank you so much for joining us on the podcast. It's great to see you again, and congrats on all your success.
Thank you so much. Thank you so much. Take care.
Thanks for watching today.
If you like this video podcast, make sure to subscribe to our YouTube channel for more cool neuroscience.